Coxella burnetii based abortions. Coxella burnetii, of course, is very highly zoonotic in nature. It can cause the most predominant clinical manifestation, the Q fever, Queensland fever, or query fever, or abortar fever in the human beings. So this particular illness has been discovered, especially in 1937. But of course, abortion or stillbirth caused by the Coxella burnetii can lead to severe damage of placenta, and Coxella burnetii will be excreted through the placenta. Even there are many reports that Coxella burnetii can also lead to maceration or mummification of the fetus. This particular pathogen is intracellular in nature and a gram-negative bacterium, and they are ubiquitous in the environment and persist as a spore form. They are present as two phases. The one is the virulent form and other is the less virulent form. The virulent form, that is the one which is existing in the nature. The one which is being cultivated, that of course requires embryonic eggs, a certain cell culture with a different gas percentage. It requires to grow in that particular cell culture. So it is usually present as a phase 2 form, but it is a highly zoonotic pathogen, and the spore can survive for quite long years and can even carry to long distances by aerosol, can lead to havoc. So the Coxella transmission and clinical signs. So major route is through the inhalation, because in the slaughterhouse or anywhere, people, when they are cutting the carcasses, they are looking through the inspecting the carcasses. Inhalation is the most common route. Cattle, sheep, and goats, they are most clinically affected by the infection, and they act as a source for the human infections also. It can also lead to chronic infection, leading to abortion, stillbirth, or even get some weak or dead offspring in the case of sheep and goat. In our paper, there is a paper in 2019, Rajagunal and I was also part of the co-author. We found there is a metritis and mummification of fetus was also there, along with the other abortions or other thing. So we confirmed this particular paper, the presence of Coxella burnetii, actually in goat, that there was a clinically mummification has been noticed. As you can see in this particular here, you can see the elementary bodies which have been stained by modified Zelius technique, and we have used a particular trans PCR or insertion sequence 1111 IS111 PCR, the most common PCR for confirming the Coxella burnetii in sheep and goat.